Thank you for inviting me here. I have always wondered why scientists at IISc and students do so well, and I think the answer lies in the weather. It's such a refreshing change from uh, coming from Delhi heat and high humidity. Uh, I was wondering what I should be talking about because it's a general sort of lecture. And I thought I would speak on transgenic crop related issues. But then I realized that we are talking to, scientific community, and it is better if I bring in some of our research work that we have been doing over the years. And as far as policy is concerned, keep it a little bit in the background. Plant breeding, as you would all know, has basically one of the ways in which so much population increase has been fed in the last century. If you look at it, it was the nitrogenous fertilizers, Haber-Bosch process, mechanization, agrochemicals, and plant breeding, which has led to our ability to feed such a massive explosion in populations during the 20th century. Mustard is a crop with which we have some experience, and I will take examples of that to make some points at the end of my talk. This is basically a crop of the dryland areas of northwestern parts of India, grown a little bit in West Bengal and in some other parts of the country. But basically it is very intensively grown here. After the rains, whatever moisture remains, this crop is sown on that residual moisture. In many areas there is some protective irrigation also available, but it is one of the major crops of this region during the winter season. This crop has many interesting relatives. This is where Junsia sits. Nigra is a condiment crop used in pickles, etc. It's also called mustard. Campestris is some of our vegetable crops and also an oilseed crop. Oleracea contains all the cauliflowers, cabbages, and broccoli and all those. Carinata is a crop of Ethiopia, and Brassica napus is a polyploid which is grown very extensively in Europe and in Canada. It's a cool climate crop, while our Junsia is very well adapted to dryland conditions and a little bit higher temperatures. Every crop, it's a bit like engineering project, there are objectives in every crop. What is it that we want to do? And years back we did a survey for all the crops that are grown in India. I'm showing you only the mustard thing. Alternaria blight is a major problem. There's an insect pest, aphids, which is a big problem. White rust is another problem, and we want to improve the quality of the oil. And there are some new emerging diseases because this crop is not given rest. The farmers keep on growing this crop year after year. So there is no crop rotation even because there is no alternate crop available. And that has led to much more disease pressure and insect pressure than should have been there. Now, in every crop, what is it that breeders would like to do? They would like to increase the yield, and this can be done through pure line breeding depending upon the breeding system of the crop. Hybrids, you know, the corn hybrids was the major development in US agriculture, which made them, not only self-sufficient, but a great exporter of food all over the world. And stabilization breeding, whatever breeding potential, whatever yield potential you achieve, you want to protect it by protecting the crop against diseases and pests. So these are aims in almost all crops. These are specific areas which are crop specific. There will be different diseases in each crop, and diseases will never go away. So this is an area where I believe more fundamental and both applied work needs to be intensified in the country. And of course, quality is always an important parameter. How do you develop hybrids? So first small story that I'm going to tell you is about how we developed hybrids in mustard. You need good combiners. You need two parents which when they cross will give you better hybrid. It's not that any two parents you cross will give you better hybrid. So you have to search in the germplasm which are the interesting parents which will get on well together so that they give you yield increase. And secondly, almost all major crops, they have male and female part in the same flower. So how do you make a hybrid? They self-pollinate. Many crops self-pollinate. So you have to make one line male sterile. It no longer produces male gametes, so it will only get pollen from the other parent. So that is the way you can make hybrids. And there are two basic methods. There is the cytoplasmic male sterility, which is encoded by the mitochondrial genome, and of course you need restorer on the other side, otherwise it will all remain male sterile. And you have very interesting transgenic approach which was published in 1990s in 2 papers in Nature. Now this is what I mean. You have gene pools, you pick plants out of these, you combine them, and then you have to check out. People have tried to predict these things, but it's very difficult to predict which 2 parents will gel. This is a male sterile flower. The petals have been removed, and you see the male part is rudimentary here, while the male part is very well developed here. So you need to create these kind of lines, either through cytoplasmic male sterility or through a transgenic approach. Years back, when we started this program, we made an interesting observation that the Indian varieties of mustard, and plant breeders have been working for many years in breeding better lines, we found that they are very closely related. There is not enough variability in these lines, whether you look at them morphologically or you look at them through molecular markers. And somebody in Poland, when I was visiting for a meeting, asked me, do you know that mustard types are grown in erstwhile Soviet Union at that time? That some parts of Soviet Union, they grow these mustard lines. So I brought some seed, and these lines are very distant from Indian lines. So there is genetic divergence between the Indian types and Ethiopian types. And out of curiosity, when we crossed them, we found that there is a possibility of making hybrids which will yield more than the Indian types are giving us. And that is how we started with the whole program, which we are still battling with. This is the first hybrid we made based on cytoplasmic male sterility. That was also discovered by us, this CMS system. And these hybrids give about 13 to 29% more yield than the best varieties. These are regional checks, these are the national checks. Again, in the other zone, it is about 20% to 30% yield increase. This hybrid is now with the breeding companies, and they are going ahead and trying to sell the seed to the farmers to get some yield increase in mustard. Now this is a continuous activity. You have made one hybrid, but that doesn't mean that's the last word. In the last 2 years, we have tested another hybrid based on male sterility, where the seed size has increased, where the oil content has increased, and it is less prone to lodging. It doesn't fall when it's too full of fruit. So this is a process which we hope, using this male sterility, will continue for many years to come. What is the problem with this male sterility? It is stable only in one female background. So we are a little bit constrained in this male sterility that it only works with one parent. It doesn't work, it is not going to work with many parents. Of course, you can have a lot of male parents, you can change them. That's what we did here. But female side, you are stuck with only one line. So years back, we decided to work on that transgenic system, which of course was not our discovery, but we modified it, and to my surprise, we filed patents abroad. We filed patent in India, we got US patent, we got Canadian patent, we got patents all over the place for making some very vital changes in that technology. And I'll just briefly explain what we did that. This transgenic hybrid was developed in 2002. and today it is 2012. It is all stuck in some courts because of the feeling against GM crops and so on. But the technology is very simple and very elegant. This is again a male fertile flower. You see that pollen is being developed here in the male part. This is a male sterile line. And this line was developed by using a ribonuclease gene, Barnase. In fact, I got that gene from Jayant, who is sitting here, years back. And you can express it in such a way that it only expresses in the male part and aborts the male gametes. And the changes that we made in the technology, I have no time to explain those, but what I want to tell you is that it took us about 5-6 years to develop the transformation technologies. It took us all that time to express the gene properly, get the thing to work both on the male and female side. We had these two divergent gene pools. After all this work, 2002, it's 2000, the weather remained little cooler for a longer period and the seed fill was a bit better. But to cut the long story short, we can expect about 20 to 30% increase in yield by using this hybrid. And it's a very versatile system which can be used with any two parents. Now what happened in the second year was, these are the trials. Now people may say anything, but I know that these trials were looked at by the best protocols that are available globally. No compromise was made with any trial sites. They were fenced. Everything was done as per the biosafety protocols. And you can see some of these here. The Rajasthan government, and that is the target area, in the second year some noise was created that you have allowed biosafety trials of these transgenics. So I don't know, in a rush of blood, they said that the trial should be burned. So we could save it at two places, the data I showed. At the third place, this was the time of the crop coming to almost maturity, and you see all the isolation by growing chickpea there. I mean, nothing was wrong with this trial at all by any standard, but still, for political purposes, this trial was got rid of. Now, look at the irony. Using the same technology, Canada in rapeseed, 97% of their area is under transgenics, and almost 70% of their area now is under hybrids made with the Barnase Barstar system. And look at how their acreage has increased. They are the biggest suppliers of edible oils, canola oil, to the whole world. The mustard meal is being consumed by animals all over the world. Even Japan, who doesn't allow transgenics to be grown in their country, is a huge importer of both rapeseed meal and canola oil from Canada. So everybody is consuming it. It is in the food chain for many, many years, and an advanced country like Canada is selling oil all over the world, while we are importing 56,000 crore worth of edible oils in India. 56,000 crores. I keep on checking this figure because sometimes I believe it cannot be right, but it is the right figure. And our farmers are underproducing, and so they are not making money, and we are spending so much in meeting the edible oil requirements. So some improvement and better hybrids and varieties have to go to the field. We made a transgenic in 2004, which is a very simple thing. It's an antisense to FAT2 gene to improve the oil quality. And let me show you what came out of that. We have now mustard strains which have 75% oleic fraction, very much like olive oil. But it is better oil than olive oil because it has 18:3 fraction, which is very important for vegetarians. Those who eat Mediterranean non-veg food don't need this because they get this their daily requirement from non-vegetarian food. This probably on a piece of paper, it looks probably the best edible oil that has been developed anywhere in the world. And we have got still some nutritional tests are being done at National Institute of Nutrition. But a simple approach, and if I can go back to this slide, we use LOX3 system here, and there is no marker gene even in this. So you have antisense to an indigenous gene, indigenous promoter, no marker gene, and we have even difficulty in taking this technology to the field because everything is in the court and we don't know when the government and the courts will wake up and give a better judgment on the issue. One of the interesting things which we can do with crops now, using the new molecular biology tools, is to put molecular markers on in the crops and do more precision breeding. Earlier, it was only phenotype. You look at it, you have to be a very clever breeder, and breeders did a fantastic job even at the level of phenotypic selection. But with the molecular markers, you have reproducible results. You know where you are navigating in the genome, you know where your quantitative traits or qualitative traits lie, and therefore, people don't have to rediscover the wheel working independently. It is all documented, so you know what you are dealing with. In fact, you know, there has been a lot of work on model species, and excellent biology has been done on model species. With the new and next generation sequencing and new tools, almost every crop can be converted into a model system now. If we have the courage to make our research framework in such a way, we can be using now crops as a model system. Of course, you won't like to put a function to every gene that is present in mustard. There would be around 60,000 genes there because it's a polyploid. But you can go selectively and look at disease resistance genes. You can look at quantitative traits in a more, at the molecular level also in how they interact. We developed an AFLP map, my colleague Akshay Pradhan, who is very active in this area, years back. In fact, this was the first molecular map which was developed for any crop in India. But we have gone away from these anonymous markers, the AFLP markers. RFLPs are very difficult to deal with. Our recent work is all using either intron spanning markers based on intron length polymorphism or SNPs. This is now a misnomer. There are thousands of SNPs available. You can navigate in any region of the crop genome and do precision mapping because there is no dearth of single nucleotide polymorphism markers. It's a bit of an expensive game, and we hope that the cost will come down so that breeders can start using these technologies in a routine fashion. And these are some of the candidate genes which have been marked which are of agronomic value. We recently did, in the last 2 years, a lot of next generation sequencing work on some of the important lines which are of critical value, and these lines, they have given us huge number of SNPs. In fact, the Rapa paper has been just accepted in BMC Genomics. We hope that other work will also get published so that everybody can use these. And this is the latest map on Brassica juncea, which is only gene-specific markers. And of course, shown here are the Arabidopsis blocks. So we can, once we know where our trait is coming, we can go to Arabidopsis genome, which has been so well characterized, and see what kind of genes are involved for that quantitative trait or for the qualitative trait. This is only the map. I just wanted to show you. This is the latest thing we made. Just to give an example of another marathon run. We were interested in improving the meal quality. Mustard meal, as it is produced in India, cannot be fed to poultry. It is very bitter. So that glucosinolates, if we take them out, this meal could be very high value meal which can be fed to poultry, swine, and so on. And this is a biochemical pathway. I don't have the time to go through that. Most of it was worked out in Arabidopsis. So model species also help. And because Arabidopsis is a close relative of Brassica species, so it's pretty useful to fall back on that knowledge which has been gathered in Arabidopsis. We started on this work in 1999 and went through a very elaborate backcross breeding program to take these low glucosinolate genes from an unadapted variety into the best Indian variety, Varuna. And we did not succeed in it. There are two things. uh, for the common, from the common sort of perspective, there are two things which are enemies of plant breeding. One is that character is controlled by many genes. So you have to deal with large number of genes. And the segregating population, your progeny will go into tens of thousands if you want to select for all the proper combinations in one go. The other problem is something which classical breeders know, is called linkage drag. Your good gene is sitting close to some bad genes. So when you transfer good gene from the unadapted germplasm into your varieties, the bad genes are also dragged along. So after this backcross breeding, we can only constitute 86% of Varuna genome. 14% was still from Hira, and that was creating trouble. We want good genes from Hira. We don't want the baggage that those good genes bring with them. Now, how do you deal with this? We know that 4 genes, 4 loci are involved in giving us very low glucosinate levels, from 120 micrograms per gram of defatted tissue to 4 to 5. Now, how do we stack these genes? Because the intervals, the mapping intervals are too large, and a lot of garbage is being brought from the unadapted parent into this along with the good genes. And this is again very elaborate process by which we picked recombinants to remove this extra genetic load or genetic drag, and we had to go through repeated cycles. Earlier it was about 190 centimorgan of Hira. It was turned into 71. We still found that there is 15% less yield in these lines. So now we have more markers in these areas, and based on these markers, each locus is going to be cleaned up for the drag problem, so that we have this gene and very few of other genes which hopefully will not affect the yield potential of the crop. Because we cannot go for lower yields. We want quality, but we don't want to compromise on the yield potential of the plant. So this was just all the strategy. What we have done now is reduced it to about 11.3 centimorgan and hope that these lines will be as productive as this. Where are we going from here? This was the first map that we created through double haploid lines. I don't have the time to go through this particular technique, which was actually discovered in University of Delhi by S.C. Maheshwari and his colleagues, but has never been used extensively for doing any technology development or product development. But anyway, this is Indian line, this is an East European line, and these crosses of these lines and mapping in these lines has given us huge amount of information, as I showed you in that molecular map that we have made now with SNPs and with IP markers. But what is very critical here is we have germplasm which shows better oil content than this. Like here, the best parent is 41% oil. In this particular cross, one of the good parents is 45% oil. Then you go to another cross. There is another line which is 48% oil. So the current work is, we have mapping population in all these, we have all the markers. So the work which is going on at present is to map all these traits in these populations also. And how will that help? Because we want to get the best character from the best line introgressed into our material. Let me try to explain it with this. This is QTL mapping, quantitative trait locus mapping in Varuna Hira cross. And we know that 4 QTL are critical. And these 4 QTL are in the Varuna Hira cross, which has only, let's say, for seed size, a particular seed size. But we want to go to a bigger seed size. So is it that there are more QTL in that, or is it that any one of these QTL is giving higher level of value? If we need to go to the highest value QTL, so that when we do the back crossing and introgression from one to the other, and all the effort we put in, we go to the best locus, the highest performing locus available in the germplasm. So that's the current challenge right now. So there is very interesting, of course, none of these algorithms have been developed in India, but a lot of work has been done in Western countries on making these map making algorithms, meta-analysis. If you have 2-3 populations, you can do meta-analysis on these and you can reduce the interval. Here it is, the QTL is falling in a very large interval. How do you transfer this large interval? It will bring a lot of linkage drag with it. But here, through meta-analysis of 2 populations, we have been able to reduce the interval. So now you are navigating in a very small portion of the genome. So the message I want to give is that this biparental process is the way to do plant genetics. Association genetics is the way in human genetics. Genomic selection is the way in cattle and in others. So each type of organism we are dealing with gives you different opportunities and you have to adopt different approaches. The biparental type process and making mapping population is the oldest technology. So maybe you don't have the possibility of looking very smart by using very old-fashioned biparental process. But the good way of going about it is, at the end of the day, is biparental process. So biparental process, multiple populations, meta-analysis, we believe will give us hold over where are the best QTL for increasing the seed size, for increasing the oil content, and for other quantitative traits. This is what is happening with the 2 gene pools. I mentioned to you that when we make crosses between these 2 gene pools, we get hybrids. The first generation hybrids we have produced are by crosses between Indian gene pool and Ethiopian gene pool. But we want to improve these gene pools. We want bigger seed size here. We want bigger oil content here, more oil content here, more seed size here. And we now know how many loci are involved. We know where they are going to map. We have reduced the intervals, hopefully, and we will be able to now improve both the gene pools to reach what I call as the 3 ton per hectare yield level. Right now, Indian varieties under best conditions yield about 2.2, hybrids yield about 2.6 to 2.8. We want to go to something like 3 tons or 3.2 tons. That is the goal. I believe now, I think because there is no bell, I hope I'm not taking too much time. I have 5 more minutes. See, like any engineering project, I was very enthusiastic about what I heard yesterday, how engineers deal with things. You have to map what you want to do in a crop. You should be clear. It's a technology development. There is some science involved because we are dealing with very complex systems. So you have to improvise as you go along. Like new markers come in, there are some evolutionary perspectives, there's some synteny perspective. You can keep on publishing papers on those because those papers are more acceptable. When you come to core breeding papers, who is bothered? Mustard is an Indian crop. If you have done a great job for India, this doesn't make an international news. Same way is our safflower, sesame. Who is bothered about these crops? Very few people around the world. When you go to wheat, rice, you know, those are global crops. Corn is a global crop. There is still more interest globally in those crops. So, in these crops, orphan crops, on which nobody is working, it is very important that we develop a roadmap. What is it that we want to do? Unfortunately, we made a little attempt a number of years back when we published what breeders are seeking in their crops and how those objectives can be met. I can tell you in mustard, aphid resistance is not available within the germplasm. So you have to have a transgenic approach because nothing is available in Brassica family. The mustard aphid has breached the defenses of every mustard crop. In fact, it is a parasite, it's a pest of the whole Brassicaceae, the whole family. So you will have to develop some transgenic approaches. Quality. Why are we so hesitant to develop transgenic approaches when they're so simple and so easy to handle? But for quantitative traits, I doubt if transgenic approach can help. How many genes can you put in? And you will have to first work out what genes are really important. And once you know, you can go through the recombination breeding also, as I have mentioned, to try to stack them into your priority germplasm. So this is not a complete picture of how we should go about. This is just based on our own experience. For example, when we started with oilseed mustard, we need germplasm, we need in vitro protocols for developing transgenics and double haploid, we need breeding strategies. You cannot get up one fine morning and say, I'll cross this with this. You have to be very selective in process make, otherwise the number of crosses will be so huge, you will be just swamped by the number of crosses in the progeny you produce. So all this requires deep thinking. And as I said, there are two major gene pools. That was an important thing which we found for this crop. Pollination control methods, which I have mentioned to you, are already in place. So the first generation hybrids are already there. The transgenic one is not able to go to the farmers because of our political and other issues. DMH1 is already with the farmers, and DMH4 we hope now will beat DMH1 and will be a more popular hybrid. What is the second generation hybrids we want to make? That will contain low glucosinolate and zero erucic with that quality traits that I mentioned, high oleic fraction and so on. A very important area for which I don't have the time, so I'm not going to go into this, is disease resistance, where we have found solution for white rust, number of genes available which can be introgressed through marker-assisted breeding. We published some of the work also. But Alternaria blight, there is nothing available in sexually crossable germplasm. So we have gone to Arabidopsis. Now tomorrow, if we transfer any genes from Arabidopsis, and we have found loci which confer resistance to Alternaria blight in Arabidopsis, we transfer these to Brassica species, and all anti-transgenic people come out and say, no, this should not be done. It is plant to plant, very closely related plants. So, and there is a whole history in wheat, for example, where genes have been transferred from wild species into wheat with great difficulty. 20 years work. In fact, sometimes when I read about this kind of work, it baffles me how these people had so much patience to remove that linkage drag, because you are bringing gene from a very wild source. The gene is very important, but it is linked to huge amount of junk which you don't want in your cropland because it depresses the yield. So now we have easier tools, and science can only, and technology development can only progress if we are willing to say that, you know, we will strengthen our biosafety system, but if there are no biosafety considerations, we will allow these kind of materials to go through. But I think technology planning, mapping needs to be done for every crop in India. And the fourth generation hybrids, of course, I mentioned to you, we have already into marking the QTLs in narrow intervals, and I think this QTL mapping will lead to even further increases. So this gives you a picture of what are the intentions, whether I fulfill these intentions or some of our other colleagues do it, it doesn't matter, but the show must go on in terms of breeding. I must acknowledge my colleagues, you know, some of them have worked with me for almost 20, 25 years. I think this is a miracle, because in India, teams seem to fall apart very quickly. Akshay Pradhan has been leading the breeding side, and he made the first map actually for any crop in this country. Arundhati has been mostly doing tissue culture work, Vibha Gupta handling back libraries and a lot of mapping work. Pradeep Verma has been working, we have been working together on transgenics. Sodhi has been looking at the field work in a big way, and a lot of these PhD students who did mapping work or developing transgenics and then pushed off. Some of them now, on the basis of the genetic stocks that we have created, like Priya, they want to study the molecular mechanism of white rust resistance. I hope they'll get money to do some more basic work on this. We are happy to provide the genetic stocks and do the sort of upstream work. I want to take this opportunity to dedicate this as a very humble dedication. years back, when I was struggling to put this program together, one person helped us in a big way. Dr. Kurian gave us 15 years back a 3.5 crore grant to develop a breeding station and breeding work and so on. Without this kind of support, DBT has been helping us, other agencies have been helping us, but 3 years, then you are lost. This kind of work cannot be done without putting together centers of excellence. Technology mapping, interaction with best of the experts abroad. We try to avoid that. What are we hiding? We are working for our own country on our own crops. But if some information, if some knowledge comes from outside, there is no harm about that. In fact, we, as I said, we are utilizing the Arabidopsis genome data. We are looking at the Brassica napus data. So even if you work on a crop which is only grown in India, there are many sister crops and sister plants whose advisors can help you. So I hope that there will be a policy of creating centers of excellence for plant breeding. It is no good for this country of 1.2 billion to only seek 4% annual growth in agriculture. 55% of our people are still dependent on agriculture, and I don't see the numbers coming down. The percentage may come down, but the population is also increasing. So thank you for giving me this opportunity to speak to you.
Thank you, Professor Benton, for a very illuminating lecture marrying traditional plant breeding with molecular technologies and bringing to the forum the crux of the matter of how one can attack issue which is both scientifically exciting as well as socially relevant. I think this is a television talk. If you accept questions.
Yeah, I'll be glad to.
We'll leave that.
I mean, outside, you mentioned about this transgenic seeds and problems related to it. So suppose you make a good seed, can you go back to classical breeding method and try to achieve that by many people working together? So that is, in the lab you make a transgenic mouse or some seed. Can you go back to the classical method and work vigorously and reach the same thing? Is it a meaningful question?
Yeah, I take your point. What you are saying is that if we push away the transgenic technologies and try to do everything through the classical breeding using marker method.
Learn from
You use transgenic technology to prove a point, but then you
No, but things go the other way better. You have found a gene in the wild germplasm, and you are going to spend 10 years in cleaning up the regions around that gene. So the smarter technology is you take out that gene and just put it into your crop. No,
But in view of the existing political, let's say.
No, but the point is, you see, tell me one young person when he knows that there are two opportunities, or he or she knows that there are two ways of doing it. One is 1 year and the other one is 10 years. And you try convincing that person, and no publication even at the end. There is a huge crisis here because biological sciences as they go are very publishing oriented areas. So the scientist, the guy will say, or the lady will say, why waste my time? So I think it would be a mistake, and this is going to soon break down, because I want to make one more point. All this genomic information that is available with us, with every crop sequence, are we just going to sit with this data and sit paralyzed and say we cannot do anything about this data? What we need to find out is the smartest ways of utilizing this data and compressing the breeding cycles. That is where the challenge is. This political challenge, I hope, will go away. But it is still long-winded, no? It is still long-winded. It's still, yeah, it's long-winded.
So how much of a complication is polyploidy and all these breeding genus, genome assembly and other things? How does it affect?
We have basically done transcriptome work, but Brassica juncea is a polyploid, is an allopolyploid with two genomes, as I showed, and its two parents are triploids. So every gene is represented almost 3 to 6 times in depending upon whether you work with basic diploids or with tetraploids. But I think we have been able to make markers which is SNP for differentiating between the paralogs and also homologs and also marking allele specific markers. It is little time consuming, but it can be done. And you know, Rapa has been sequenced by Chinese, Nigra has been sequenced by Canadians, and Oleracea has been sequenced by Chinese. And we are hesitant to use even that knowledge. So sometimes the picture looks too grim.
Can I make a comment? I'm neither an agricultural scientist nor even a plant molecular biologist, but I burned my fingers once on transgenics. And I'm sticking my neck out and again trying to burn my fingers. Ultimately in these processes, all rational discussion we should be sensitive to, but eventually there is no alternative to fighting and defeating in the long run anti-science Talibanism.
Yeah.
There is no other alternative. At some point of time, we have to stick our neck out. It's a long-drawn process without sacrificing what we call logical arguments. There might be problems with, even later, Nature there's an article, there may be problems in different issues. But this mindless anti-science Talibanism should be fought and defeated.
I agree with you. I just a small comment I want to make, Professor Vijayan, that you know, it's not like pitching one person against the other. I think we very badly require scientific evidence. We should be committed to good biosafety protocols, but we will be missing out on a lot of science. And second small comment I want to make is that, you know, next 20 years are the most critical for India in terms of food. After that, what happens? You know, if somebody is projecting, I am doing some science which will help after 20 years, I think there are big problems with that. We need to deal with what is available right now. So that's why I'm making this point that we should do a technology mapping. What is available abroad? Can we buy it and put it into our crops? Why be so hesitant? The farmer is waiting for more productive material, you know, and we have to fulfill that requirement.
Sir, thank you for giving such an eye-opening lecture. And I always wonder why plant produces something which itself do not need. Say, for example, oil, laxatives, medicines, which plant itself do not need. So why some plants they produce oil?
Storage material.
Yeah, that is basic thing, but
Storage
how it is
material then
useful for
needs
the plant itself?
seed needs to germinate, so it needs some energy. Either it can come from fats or from carbohydrates, you know. So that's very simple.
Thank you, sir.
So if there are no further questions, I want to thank Dr. Bender for giving us.
Let us thank Professor Usha Vijayaraghavan for chairing the session and Professor Pentel again for a